Hello, good afternoon to everyone who's listening to our talk. My name is Ulrike Scheffler. I'm heading the business development department at FyoniBio, and I would like to present today how we can boost your commercial success with our customized cell line development and tailor-made bioprocess platform. Shortly, to introduce FyoniBio, for whom FyoniBio is still a bit unwritten leaf. Some of you know us probably already. FyoniBio was officially founded 2 years ago. We just celebrated our second birthday. FyoniBio is located in Berlin Buch at the Science Campus Berlin Buch, which also reflects our nature. We are a very science-driven CDO, soon-to-be CDMO company, offering stable cell line development, process development, analytical development, and characterization services. We are currently 35 employees, but are expanding, not only personnel-wise, but also when it comes to our technology platform. FyoniBio was founded 2 years ago, as I mentioned, as a spin-off from Glycotope. Glycotope is an antibody discovery company who did all the CMC part completely autarkically by themselves and sent 3 of their pipeline candidates into the clinic. So there was a strong CMC expertise implemented at Glycotope, and this CMC expertise was 2 years ago transferred to FyoniBio, which allows us now operating as a completely independent company, offering stable cell line development, process development, analytical characterization with a very comprehensive MS-based platform. We have our own bioassay department, which is able to set up all needed potency assays. And this is something where we really stand out from other CDMOs. We act as a clinical laboratory and offering clinical bioanalysis services to analyze clinical samples regarding PK and ADA analysis. So we grew into soon to be a full-blown CDMO here. On this slide, you still don't find any GMP manufacturing capacities. It's not here yet, but we are very close to be able to offer the entire CDMO value chain to GMP manufacturing. All our services are ISO 9001 compliant. Today in this talk, I would like to focus on our cell line development and process development platform. You know, every clinical project, of course, starts with a molecule, but in order to be able to produce a molecule, you need to have a good producer clone available. And here we also stand out from other CDMOs, since we not only have one CHO-based platform, we have a CHO-based platform, this is the topic of this talk, but we don't only have one CHO-based platform in place, we have our human cell line development platform as well established, GlycoExpress it's called, and this would be the platform of choice if you have a very complex glycoprotein which cannot be produced in CHO. We use this platform to generate clinical producer clones for clotting factors, factor 7, FSH, and surfactant D. But this GlycoExpress platform is not the topic of this talk today. I will focus on our tailor-made platform. Tailor-made, as the name already indicates, these platforms allow us, so our clients, to produce their molecules very explosively. And here the tailor-made platform also is not only composed of one host cell line. Now we have 3 host cell lines integrated in this platform, CHO-K1, a CHO-GS system, and CHO-DG44. And this versatility allows our client actually to test different cell lines to see which one is most suitable for their particular protein. And this is of crucial importance if it comes to biosimilar development. We have seen that each and every host cell line generates slightly different PDM and glycan structure. So here it's of great advantage if you can test in the beginning of your project as many host cell lines as possible. So we have a CHO-based platform. CHO cells are the proven expression host for numerous clinical and commercial approved proteins, as you know. It can be used for all kinds of antibody-based projects, bispecific antibody projects, biosimilar development, FC fusion project. So basically, I mean, especially us coming from a glyco company and having all the expertise and knowledge around complex glycoproteins, this platform is also offered, of course, for your recombinant glycoprotein. The platform does not only consist of different host cell lines. We also integrated a very strong vector system with strong promoters, allowing us to reach high expression rates. And these high expression rates are even further elaborated by using a transposase system. And all these different components together allow us to generate producer clones with expression titers of up to 10 grams per liter. And in addition to the 3 different host cell lines, we generated our own CHO flow cell line. What is CHO flow? CHO, it's the whole name of the host cell line. F for fucose and low for low. So we have a cell line which produces not only low fucosylated proteins, actually a fucosylated protein. It's a FUT8 knockout cell line, which can be used to produce afucosylated molecules with enhanced ADCC effector functions to significantly enhance cell killing potency of your antibodies. So these are the short introduction to our tailor-made platform, including our CHO flow cell line. Of course, we have this versatility available of different host cell lines, not only with the tailor-made, but also with GlycoExpress. And this gives you as a client also the possibility to test all these different cell lines in a feasibility study to select the most suitable host, not only in respect to titers, more of importance to select the clone which produces your molecule at the desired quality. Now I would like to come to a case study here to show how the versatility of a cell line development platform in combination with tailored process development approaches leads to the generation of highest expressing clones and for you to get commercially viable manufacturing processes. And for this case study, I would like to invite you to our Berlin famous TV tower tour across the different stages of a CMC project, starting with a generation of stable pools over the clone level stage till the finalization of a high production process. And this case study using our elevator story is based on a client's project, client's bispecific project. The client is Strike Pharma, a Swedish company, which has a very, very promising clinical molecule in development for immuno-oncology applications. Basically, actually, it's kind of a vaccine approach using an fusion antibody. It's a bispecific antibody, which is C-terminal fused to an scFv, which then is bound to a peptide, which represents a T-cell antigen. And the T-cell antigen is basically the vaccine, which is then delivered through the antibody into immune cells, monocytes. We appreciate very much that we are allowed to show our clients Strike Pharma's data. The molecule was transferred together with this information that there are challenges to be expected in early stage production. In early stage production approaches, they have shown that the productivity could be rather low. They have seen that it tends to aggregate and eventually some challenges in the protein's stability could be observed. So we like challenges, so we approach this challenge with our versatile strategy to generate high producer clones. It all starts, of course, at the transfection of the host cell line. And here we already set up a multi-parameter transfection strategy in order to generate versatile pools. For transfection, you use different parameters, different ratios of heavy and light chain of the antibody encoding vector. We use different promoters. We tested different selection pressures. We incorporated different signal peptides. We tested transfection of different amounts of DNA. And we, of course, also included our transposase gene integration system. In total, we generated 24 pools. And as you could see, the pools showed quite differences in the expression levels. The highest, most promising pools showed pool titers up to 2.5 gram per liter. 4 of the best pools were then proceeded with single cell cloning. I mean, of course, first we tested also the quality. We have seen that the generated produced material from the pools have very promising monomer content, so we actually don't see a tendency for aggregation, and the quality, the integrity of the molecule looks good as shown in the SDS gel. Could clearly see a nice clear band of heavy and light chain. So 4 best pools were then proceeded with single cell cloning. Here we also implemented a very smart screening procedure. All starts with using the image-based system, the cell selector, which allows us to screen several thousand clones. The best clones were then proceeded in a expression static titer measurement assay, and the best clones we identified in this static expression assay were then proceeded in a fed batch screening round using the EMBER system. And here we got the winning clones with titers of 4.4 gram per liter. Could see we actually could double the titer from the pool stage to the clone stage. 6 of the best clones were then proceeded in a next level of cell line development. Cell line development doesn't only mean we screen for best producing clones. We, and this is more of importance, want to show that the clones can be manufactured well. And we need to check and compare the different clones regarding their manufacturability behavior, and also see if we can already during cell line development adjust a bit of the upstream process development conditions in order to improve titers. So in the first step of our cell line development integrated upstream process development approach, we tested different media and feed strategies and compared the expression rates of these 6 different clones. And as you see, already the first step of our early process development approach caused a titer increase by reaching 5.3 gram per liter. In a second step of the early small-scale USP development step, we tested additional process and additional bioreactor parameters. We tested different feeding strategies, different inoculation densities, and analyzed the productivity of the top clones. And by this second step, we could further increase expression titers and came close to an expression titer of 6 gram per liter with a winning strategy. And also here we can see that actually we could not only improve the titer, but also can identify that we could lower the amounts of aggregates, which were anyway quite low from the beginning, but we could further reduce aggregation behavior with a certain process strategy. So this is the end of the cell line development approach. We selected the top clone with a titer of 5.8 gram per liter, already have a good indication about the process parameters. But of course, with the winning clone, we went on in a full-blown upstream process development approach, testing various different process parameters, pH shifts, T shifts, guessing strategies on small scale runs, but also on a 5-liter upscale run. And by this very intensive screening and optimization procedure, we actually could improve the titer to up to 9 gram per liter, which I would say is one of the highest among our among also the competitors' cell line development systems. And I have spoken a lot about the upstream process development part in the last slides based on this client's case study. But of course, we also do downstream process development. This is just not the focus of this talk, but this little chromatogram and graphs shows you that we could significantly also lower the amount of impurities like aggregates by implementing a 3-step column purification step. So at the end of our process development approach, incorporating upstream and downstream process development, we set up manufacturing process reaching an expression titer of 9 gram per liter and be able to have a very, very clean and pure drug substance after the downstream part. And this process can then easily be transferred to a CMO, or as I mentioned, soon we will have our own GMP manufacturing facility, then can be processed in our own facilities for the production of the clinical material. So now I'm already at the end of my talk. I hope I could convince you that the approach of a versatile transfection strategy leads to pool diversity, and a diverse pool strategy allows us to increase the probability to generate high producing clones. Our cloning strategy intelligent design resulted then later in a 2-fold increase in titers from pool to clone level. The customization of the upstream process development leads to superior and commercial viable production processes. And all in all, at the end, we delivered a process which is of total satisfaction of our client and leads to their commercial success. So the combination of having a choice and customized platform processes is a key to success. Thank you very much for listening, and I hope you still have very good time here at the BioPet Forum listening to additional interesting talks. Thank you for listening.